Hello and welcome everyone. This is the first part of, I believe it's a three-part series on cellular level anatomy and physiology, and we'll just go ahead and get started. Over the course of this, we're going to look at the different parts of the cell. We're going to explain their functions. We're going to hone in on the ones that are most important for you for the paramedic and kind of, set the others to the side. We're going to describe the types of active and passive transports, and we're going to explain what cellular respiration is. So we're going to start out, like we said, this is cytology. We're looking at the study of cells. And the big things to remember, of course, is that cells can vary in shape, size, how they function, but they do actually share some very common traits. They have a nucleus. All cells have a nucleus with the exception of the red blood cell. They have organelles, they have a cytoplasm, and they have a cell membrane. So they have little organs that float around inside them. They have a membrane that kind of keeps it all stable and together, and they have fluid that those membranes sit in, which is known as the cytoplasm. So this is just a typical picture of a cell, just a cell rendition. But we need to know that the cells are the smallest functioning units of life. Now, this is different than the atom that we talked about before. Cells are produced only by the division of pre-existing cells, and each cell has to maintain homeostasis. It has to be happy in order for it to survive. And our cell membranes have to be nice and healthy to separate the cell contents from the surrounding fluids. It's very sensitive to the environment, and it provides that structural support, just like, if you think about us as an organism, our skin, our musculature, and our bones, they're kind of protecting and encasing what we have inside us, the organs inside us. It's the same thing with the cell. So we need to know how cells get what they need in order to survive, and that's how we talk about cellular transport. So there is a thing called vesicular transport, and in vesicular transport you have a thing called endocytosis, and that's where you're taking extracellular fluid and you're bringing it into the cell. And it does this through, it's called pinocytosis. It's cell drinking is literally what that means. Pinocytosis means cell drinking. And you can see that cellular membrane. Let me get my highlighter up here so you can see. You can see with the, my highlighter is not working. Why is it just sitting there and staring at me? That's a little weird. Okay, so it's not working right now. That's okay. But you can see with the cell membrane, it's actually comprised of two layers, and they kind of face each other. So it almost looks like this. And that is that outer cell layer that's kind of the cell wall that's protecting the internal contents of the cell from the external. So in pinocytosis, it creates this little pocket that the fluid can sit in, and then it comes in and it pinches off, the cell wall pinches it off, and then that allows it to be brought into the cell in this little vesicle, because it created the vesicle as the result. Now, the other type of endocytosis is called phagocytosis. So remember, endocytosis is bringing something into the cell. We can do it through cell drinking, which is pinocytosis, or cell eating, which is phagocytosis. And it's the same concept. That cell wall is creating this little pocket, it's picking it up, enveloping it, it's pinching it off and bringing it inside a vacuole. Now on the flip side, when the cell is done with whatever it needs to get rid of the extra products or waste products, whatever it is from that cell that cell should generate. So it does that through exocytosis, because we're exiting the system. And it does it in this very similar fashion. You have this little vesicle that's filled with whatever the contents are, and it will go to the cell surface, and it will create that opening and allow it to exit. So that's exocytosis. Now we're going to kind of talk a little bit more about cell wall permeability and how it allows certain things in and certain things out. So this is beside the concept of what we've learned so far about endocytosis and exocytosis. So we're going to look at how the cell transports contents in and out as well. Hopefully I have kind of tweaked around with this laser pointer, and I'm hoping that it will work. Yes, it is working now. Okay, very good. So let's move forward. Okay, so the cell wall is considered to be semi-permeable. It will allow some things to enter freely or leave, but it will restrict the passages of others. So permeability is considered or defined as the rate at which a substance can pass through the membrane. I'm going to go ahead and pull up my pointer so that we can kind of take a look at this. You do have these channels. You have this channel here. This is considered an open channel. That's what allows things to freely float through. But then you also have this other channel over here that's gated. It is closed, and it requires a key to unlock the door to allow whatever contents are coming into the cell. And that's what we're going to focus on. In the previous slide, I mentioned that cell wall is made of two layers. This is a phospholipid bilayer, so you have these little tiny phosphate balls that have two little diglyceride tails, that attach to it. So these are fatty acid tails. So that's how come, if you ever think about water and oil don't mix, that's how we are able to, look at this as being semi-permeable. It will allow certain things in and other things it will not. So that's how we kind of regulate that. Let's take it forward a slide. And this is a very similar picture to what we just looked at. There's a lot of things. If you look at a cell wall, you have little glycolipids, little cholesterols that are kind of sticking out, different things. You have different proteins, and each have their own specific job. But we're going to focus on what we talked about with the channels. So cellular transport, you're moving contents in or out in one of two ways. It's either going to be a passive process or an active process. So anytime you think about passive, you need to think about this. This doesn't require energy. It just happens. It's a passive process. No energy is required to make this happen. It just occurs. Whereas active transport does require energy, and energy is in the form of ATP. So let's kind of dig into the types of passive transport that we see. So with passive transport, this includes, and remember, this does not require energy. So we're talking about diffusion, osmosis, filtration, and then you have a condition called facilitated diffusion. So let's look at each of those individually. With diffusion, it's the most common means of passive transportation, and you have high concentration moving into a low concentration. They always talk about taking a sugar cube, dropping it in water, and then watching as it slowly dissolves until all the particles of that sugar cube are evenly dispersed throughout the water. That is what we're looking at with diffusion. So if you think about it in terms of this, if you vomit, you're bringing up the secretions from your stomach, but you're also having to backfill what fluids were necessary and lost as a result of that vomiting. So then you'll have those fluids passively diffuse to reconvene where you lost them. So that's a type of diffusion. You're allowing that to diffuse, and that's how people end up getting dehydrated if they've vomited for extended periods of time. Now, osmosis is a little different. We're going to spend a little time on it in class because I want to make sure everybody's comfortable with this particular topic, because it sometimes catches people up, and we're going to spend some time here. But with osmosis, this is not talking about, remember when we talked about the sugar cube, we were talking about the sugar cube being dispersed evenly through. In the case of osmosis, we're not talking about the solute. We're not talking about the sugar cube anymore. We're talking about the water that the sugar cube is in. So in the case of osmosis, the water will flow from low concentration to high. It's trying to evenly disperse out the contents. And you'll see in the picture here, take a few minutes, pause the video, take a look at it. What you'll see is that high concentration of albumin, which is a very large protein molecule that is kind of packed up in here, and you have water over here. But if you look at the little solutes, those little pieces of albumin, they're more spread out over here. So the water is going to suck through that little semi-permeable membrane, and the goal is to get the same distance, if you will, if that makes it work for you. You want those particles to be spread out evenly, just like they are on the other side. So that's how that water comes across and becomes diffused. Now, I have the picture of the snail up here because if you were ever a mean little ornery kid and you would pour salt on a slug, what you were doing, that slug's skin is the semi-permeable membrane, and when you put all the salt on top of the slug, you were causing all that water inside the slug to be pulled out, so you cause the slug to shrivel up. So that was an example of osmosis, even though at 5 years old you really didn't know any better. And then you also have the filtration, and filtration, the best example of that is coffee. So when you get up in the morning, you pour the coffee grounds into the top, and then you start running water through it. That pressure is being applied to force water and the dissolved materials through that membrane, or in the case of the coffee, that's coming through the coffee filter. And then facilitated diffusion is where you're actually using a carrier molecule, and that's where we were talking earlier. We mentioned that you do have those gates that are closed gates, that in order for the contents to enter the cell, you have to have a key to unlock the gate. When the case of the facilitated diffusion, that's what we're looking at here. We're looking at some sort of carrier molecule that comes in and it latches into the site on that receptor, and it says, okay, we can allow this to come in, and it'll open the gate and allow that content to enter. So our sugar molecules are something that's a very good example. They are large, they are not able to enter the cell without help. So our carrier molecule is insulin. So the insulin is released, comes in, unlocks the gate, and allows the sugar to enter the cell. That's why we talk about diabetics and how much of a problem they have when they're faced with type 1 diabetes, or type 2, for example. But you're looking at the amount of insulin that's available in order to get the sugar into the cell so that it can be utilized for energy purposes. So that's a very good example of facilitated diffusion. Now, we mentioned that active transport requires energy. This is going to get energy, or our cells are going to get energy in the form of ATP. So you have adenotriphosphate. So if you look here in this picture, you have adenosine and you have 3 phosphate groups attached. It's just like, I have the Kit Kat here because, the song, give me a break, break me off a piece of that Kit Kat bar. Well, if you think about it in that term, when that phosphate breaks off, just like with the Kit Kat, that is your energy source. Now, suddenly, that is no longer adenosine triphosphate because it doesn't have 3 phosphate groups. It only has 2, so it becomes adenosine diphosphate. But the cool thing to remember is that it's freely reversible. So your body can turn around and reconnect a brand new phosphate and recreate that ATP. So with active transport, this is in the case where you're moving against a concentration gradient. So you're going from low concentrations to high, and you're having to push. And an example of this is the sodium-potassium exchange pump. So this is really kind of cool in how we look at electrical conduction. So a good example would be, cardiac conduction. So we look at the heart. The heart has to beat in order for it to get the blood where it needs to go, obviously. And it does this through this idea of the sodium-potassium exchange pump. And we're going to put some time into it in class and kind of dig into it a little bit more. But you got to remember, and this comes up from time to time, particularly in class, to make it a little bit more understandable. But your sodium is primarily found in your extracellular fluid. Your potassium is primarily found in the intracellular fluid. So potassium inside the cell, sodium outside the cell, and that exchange is going to require a release of energy for those two to pass. And that's how you get that electrical impulse that you get with the cardiac pumping. But we will spend some time on that when I can talk with you guys directly and make sure everybody is on the same page. And that is it for today's lecture, or at least this section. If you have any questions, let me know.